Hi everyone, and welcome to our review on dihybrid inheritance. So what we're going to be doing in this video is having a look at the specification reference 6.1.2b part 1, genetic diagrams to show patterns of inheritance, focused on that dihybrid inheritance this time. First thing we need to understand then before we delve into this topic is a couple of definitions. First one, gene locus. This is where we're talking about the actual position of a gene on a chromosome. So wherever you see gene locus or gene loci, it's always referring to gene positions on chromosomes. Second word, dihybrid. Now, di just means two. So dihybrid inheritance is one that involves two gene loci. So when we consider when we'd be looking at this whole concept of dihybrid inheritance, then this is where we're looking to see this simultaneous inheritance of two characteristics. So last time we had a little look at the monohybrid inheritance, where we're looking at individual characteristics being inherited independent of all others. This time we're looking at those two characteristics being inherited simultaneously, so at the same time. If we consider our little independent assortment that occurs during meiosis. Now, what we've got in our little diagram here then are a couple of sets of chromosomes. So we've got homologous pairs of chromosomes, and what we're going to do is we're just going to label up the actual alleles present. So these red ones are going to have capital A's, these ones are going to have lowercase a's, and then just for simplicity, the blue ones we're going to have the B's. So top one, two uppercase or capital B's, and the bottom one, two lowercase b's. Now, what we're going to have a look at then is what happens during this process of independent assortment. So basically, how are these two different genes, the A gene and the B gene, going to actually end up in those gametes? And what we see is this. So let's just get these back on again very quickly. So these are going to split into four potential gametes. And just because we are doing gametes, we are going to stick them in a circle, circle-ish anyway, it's not really a circle. Anyone who does maths, please don't criticise my shape naming. Now, what we're going to look at then is the potential combinations of those different genes in those four gametes. So our first one here is going to have one of those capital A's, and it's going to be one of the capital B's, because it had taken one from the first chromosome and one from the first chromosome on the right. The next one is going to be one of our lowercase a's, and that one might have combined with one of the capital B's. On the other side, we can have a capital A and a lowercase b, and then we could also have a lowercase a with the lowercase b. So if we look, we've basically got four different potential combinations with our gametes. So if we just rewrite it a little bit clearer, we've got this one, this one, this one, and this one. So basically we've got both dominant, one recessive, and then the other way around, and then both recessive. Okay, so let's have a look at an example of a dihybrid cross then. So what we'll do is we'll go through the two different ways that we can actually set this out. First one, obviously using each individual line to show it, and then we'll do the Punnett square. So what we're going to be doing in this case is we're going to be having a look, obviously, at two of these phenotypes. So in this case, we're going to stick with the pea plants that we looked at last time, and we're going to start off with some yellow and round seeds. And they're going to be crossed with green wrinkled seeds. Okay, so there are two phenotypes, okay, two characteristics, yellow or green, wrinkled or round seeds. The next thing we need to do is have a look at those genotypes. Now the genotypes in this case for yellow, because we are starting with our parent generation here, we are going to have both capital Y's for our yellow and both capital R's for the round. That's then going to cross with the recessive characteristics. So we're going to stick to lowercase letters, two lowercase y's for the green and two lowercase r's for the wrinkled seeds. Next thing, we need to split those gamete genotypes up. So because obviously we've only got capital letters on the left and lowercase letters on the right, that means that we are looking at two potential gamete genotypes, which could be this or this. So both lowercase or both uppercase. They're the only ones that we could potentially have as the gamete genotypes here. Then what we need to do is have a look at that F1 genotype. Because these are being inherited together, that means that all of them are obviously going to have one capital and one lowercase y, one capital R and one lowercase r. Now, because of this, they've got one dominant allele in each case, so their F1 phenotype will be yellow and round. And all of them will be yellow and round seeded. The next thing we need to do then is have a little look at splitting these down into the genotypes of those gametes again. So remember, our F1 genotype is one capital and one recessive in each case. And that's going to be crossed obviously with a capital and a recessive for each trait again, because we're just crossing two of that F1 generation. Now, when we look at the potential combinations of gametes here, we could have both capitals, we could have a capital Y with a lowercase r, we could have a capital R with a lowercase y, and we could have both lowercase letters. So those are the four potential gametes that we could have here. Now at this stage we're going to go to the Punnett square because this is by far the easiest way to do this. So male and female, obviously remember the female is the vertical, the male is the horizontal here. So what we're going to do is we're going to fill out our gametes. So remember we've got our four potential combinations. There we go. So we've got both capital, both lowercase, and then the alternating capital and lowercase in each case. Same thing as we go down here. And we've got lowercase and an uppercase, and then both lowercase. So this gives us all the potential gametes in both the male and the female population. Then we do the same thing we do with every Punnett square. Whatever is at the top of this column goes into the squares beneath it. Now you'll notice this one is slightly larger than our usual Punnett square because we've got four potential gametes. Therefore, we need to have a 4x4 grid for our potential offspring, 16 potential boxes. So we've got a capital and a capital, so we can literally just go down and fill all of those in first of all. We can do that for all of the different columns there. So we've got a capital and a lowercase, capital, lowercase, capital, lowercase, capital, lowercase, lowercase, capital, lowercase, capital, lowercase, capital, and then finally both lowercases. So I always suggest that you start with this very logical way, and you'll notice how I'm splitting my Y's and my R's up. And that's just because it's a little bit easier on a quick glance in an exam setting if you've got both Y's and both R's next to each other. Next thing we're going to do is go across, so we've got capital and capital. So in each case we need to just fill out that same thing. Then we have a capital and a lowercase on our next row. And it doesn't matter which way round you put these at all. So if you are going with the capitals in one place, the lowercase in another, it really doesn't matter. Just put those letters together. So the Y's together and the R's together, just for simplicity when we're going to come to actually work out what the phenotypes would be. Trying to do that and talking is not an easy thing to do. Okay, so hopefully I've not made any silly errors there. This gives us our 16 potential combinations of the offspring's genotypes. So the next thing we need to do is basically work out what colour and if it's round or it's wrinkled. So I'm actually going to use different colours for this for simplicity. Now, what we do know is that yellow is the dominant and round is the dominant. So in this first box in the top left, we've got obviously capitals of each. So that is going to be a yellow round. We've got yellow round here, yellow round, and yellow round at the bottom. Next one along, we can go yellow round there, yellow round here. You'll notice I'm missing a couple because they don't have the capital R, therefore they won't be round in those cases. We can have one here, that one's a yellow and round, and we've got yellow and round there, and that's all the yellow and rounds. Next, we need to do our yellow wrinkly ones, just to save me changing my colour pen. So a yellow wrinkled would be a capital or two capital Y's, but only lowercase r's. So first one we've got is this one. Oh, so this is going to be a yellow wrinkly one. This is a yellow wrinkly one, and we've got a yellow wrinkly one up there. Then obviously we're going to go to the greens because we've got no more capital Y's left, sorry, in any of the boxes that we have not labelled yet. So two lowercase y's, two capital R's means a green round, and we can do green round here and here. This just leaves both lowercase y's, both lowercase r's. That's a little green wrinkly one. Now, if we do a simple little counting up, then what we can see is that we've actually got 9 of these are going to be yellow and round. We then have 3 of them which are going to be yellow and wrinkled. We've got 3 that are going to be green and round, and 1 that is green and wrinkled. So what we can see here is that we've actually got our little ratios, again, out of 16 in each of these cases. So that's 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16. So we've got our fractions here. And what we need to remember is that if these traits are being inherited independently, then we can predict the outcome of these crosses. So basically what we find is that each of these individual traits can be treated as a monogenic inheritance. So 3/4 of them would be the dominant trait, 1/4 the recessive in that F2 generation. So in order to work out the probability of both of those traits being inherited, we just multiply them together. So the 2 dominant traits would be 3/4 times 3/4. So hopefully your maths knowledge is up to scratch. If not, the joy is we can use a calculator, remember folks. But that does give us 9/16. Think back to what we just saw, those ones that were yellow and round, the 2 dominant traits, 9 of them out of the 16 were there. So this just gives us our probabilities. So remember, yellow and round are the dominant traits, so 3/4 times 3/4, 9/16. Yellow, dominant, wrinkled, recessive, so 3/4 times 1/4, 3/16. Green, recessive, round, dominant, 1/4 times 3/4, 3/16, and then 2 recessive traits, 1/4 times 1/4, 1/16. And this gives us the all-important ratio that we saw in our example, 9 to 3 to 3 to 1. So remember, that 9 is 2 dominant traits, the 1 is the 2 times the recessive traits, and those 2 3s are basically 1 dominant, 1 recessive. As always, I hope you found this video useful, and I do suggest that you subscribe to the channel so you can see when the next video is uploaded. And don't forget to head on over to the website where you can find an array of different resources to help you with your A-level biology studies.